Professor Ada E Yonath, Nobel Prize in Chemistry for her studies on the structure and functions of the ribosome. She obtained her PhD from the Weizmann Institute of Science for X-ray crystallographic studies on the structure of collagen. Currently, she is a member of the United States National Academy of Science, the American Academy of Arts and Science, the Israel Academy of Science and Humanities, the European Academy of Science and Art, and the European Molecular Biology Organization. It's indeed a great honor to have you with us today. We welcome you, ma'am.
In English, okay? For those of you who have not attended earlier, I just want to let you know, Professor Yonath, when she was 5 years young, she was very curious. She got interested in science, and in order to learn science, she broke even her arm because she wanted to measure the height of her building by not using a barometer. Then, of course, she got interested in chemistry. No doubt she has won a Nobel Prize, but the reason she won the Nobel Prize is because she loves science. Because of her love for science, she dedicated herself, and it was the perseverance and the hard work that she put in over almost 50, 60 years that has earned her the Nobel Prize. For the last 3 days, we have already heard her speak about her research, and today she's going to talk about her research in science, how it has impacted medicine. Her research in structural chemistry has enabled us to create new antibiotics. In the morning, we heard Professor Harosh talk about quantum physics, how it has enabled us to bring out mobile phones, to bring out lasers, to bring out computers. How quantum physics has brought us technology. Similarly, her research in science, in structural chemistry, has brought phenomenal advances in medicine, and that's what she's going to talk about. So I would like all of you to appreciate her research and give her a big hand, please.
Thank you. So I want to tell you today my way from basic science to advanced medicine. I'm a scientist, and when I became a young scientist, a young researcher, I wanted to answer questions that are still not understood in life science, in world, and so on. And I thought that the translation of the genetic instructions that are embedded in the DNA into cell workers that are called proteins is a very interesting question which was not answered at that time. So you all know what is DNA. You all know that DNA has instructions how to make components in our body. The main components are proteins, and they do almost all life activities in all living cells, in bacteria, in mice, in elephants, in flowers, in cockroaches, in fish, in everything that lives, in all cells. So I have here five examples of proteins in the body, in human body, like structural proteins, hair, skin, cartilage, signaling proteins like insulin that signals the level of sugar in the blood, transport proteins like hemoglobin that transport oxygen from the lungs to wherever it's needed in the body, like allowing this motion, which is muscle motion. Muscles are also proteins, and they need energy. It means they need oxygen. And there are proteins that are in receptors, like allow sight, hearing, and other senses. there is a class of proteins. Most of the proteins are enzymes. Enzymes can chop other molecules. So when we eat proteins, like when we eat an egg or cheese or drink milk, it's full of proteins, but they are not useful for the body. They have to be cut, and from their components, these proteins that the body needs are made. And this cutting is done by these proteins that are called enzymes. So to repeat, there are some proteins that are required for smooth life, like breathing and muscle motion, and others that are responding to pressure. And they are needed only when there is pressure, but they have to be made then, because proteins cannot sit in our body for years and years, only a few days. So when there is a heat or cold shock or immune response when a disease comes, sometimes there is heart failure that can be taken care of by proteins, or when dust comes into the eye, the tear that comes out has a protein inside that can chop the walls of every bad bacteria. So if in the dust there is a bacteria that could cause infection, it gets rid of it immediately while the eye is being washed. In nature, structure means function. It means only when the structure or the fold is maintained, function can be obtained. So what you see here are paper clips. You know what they do? They keep together pieces of paper. When they have the right structure, they can do the work. When they are unfolded, have different structures. It's exactly the same paper clips, but they cannot do the job. So fold is important. And you can see here a fold of a typical enzyme. It's made of 150 or 250 between this to this components that called amino acid. I'll come back to it. For many, many atoms. And in this very complicated structure, there is here a groove that is called active site. Here the activity is. So we look into it, we enlarged it, and I rotated it, and I looked into it with the substrate of the active site. It is here sitting in khaki, and the enzyme chops it here, and you see how well fantastically the structures are matching. If not, the action will not take place or will be wrong, and the body cannot afford wrong actions. So as I said earlier, proteins are made of amino acids. They are long chains like this. There are 20 types of amino acids. And the fold, the important fold that allows, that is carefully designed for fulfilling the function, is determined by the sequence of the amino acids. So there are 20 types, and normal proteins are around 150 to 250 amino acids, so the fold is important. And it allows the functionality. What is deciding the sequence of the amino acid of this component? The DNA, the genetic code. The DNA is a collection of instructions. It's a 4 letters language, just 4. And with this, they can have instructions for all the thousands and 100,000s of different proteins. So each triplet, each 3 of the 4 letters that are also called bases, each 3 of them are coding for a specific amino acid. So there are many combinations, many more than 20 types, but each 3 have one amino acid that fits them, or the opposite. Each amino acid at least have one triplet that codes for it. So what happens is that these are the amino acids. There are 20 types, but I show here only 5 to represent them. They all have the same backbone. And you can see that this backbone can be connected to each other. I made it different colors, but chemically they are the same. They have different side chains. The side chains give them the chemical properties. and some of them can be tools. They come together, and they grow and grow and grow, and now I didn't draw the side chains that have no activity, only those that can be tools. And then they grow even more, and here I didn't put any detail, just to show you that it's grown. And when it is complete, it can fold and have an active site, as you saw earlier. This is here the active site. So all this was known when I was a young scientist. It was known that there are amino acids, 20 types, they come together, they fold, and the sequence of them here, the order is determined by the instructions of the code, of the genetic code, but was not known is how this is happening. How? This is what I wanted to know. How this is happening? How the cell knows that it needs a protein, it knows that it wants a protein, and how does it make all these amino acids come together according to the order that is dictated by the code? So this is what I wanted to know, how. I was not the only one that wanted to know it. Many scientists wanted to know it. Many people wanted to know it. Some of them were very distinguished and very successful, yet they failed. So when I was a young scientist, there was no knowledge of this step, how. And I thought if all of the other ones failed, maybe I will also fail. Although I looked into this process in a slightly different way. I had my own curiosity-driven way to look into it, but I thought that maybe I will also fail. Yet I knew that if we make it, we will have a real big smiley. And we made it, and I hope that you are smiling with me. So what really happens? The DNA, this is the molecule, the double helical structure of DNA, has 4 letters. You can see here inside 4 different colors, purple, red, yellow, and green. They are ordered in base pairs. They are called bases, and they are ordered in pairs. And here is all the information. It means these 3 are coding for 1 amino acid. This 3 is coding for another, and so on. But the information here is buried in the very complicated structure, double helical structure. So first, it is being transcribed to a similar molecule called RNA, in this case messenger RNA, which has also 4 letters, 3 exactly the same as RNA, 1 as DNA, 1 a little different, makes the same type of pairs, like DNA, RNA, RNA, DNA, all the same. But RNA can exist as a single chain. It doesn't need to be double helical. Therefore, the information here is available and can be translated to protein. Here is a growing protein. Who translates it? There is a cellular body, cellular complex called ribosome. And this is what translates ribosomes. Ribosomes exist in every living cell. When I started, nobody knew how they work. They knew that they are involved, but not how they work. They knew a lot about them. They knew that they are complex and they are made of many components, but not how they work. This is what I wanted to. The ribosomes in all cells are universal. They function in very similar way in all cells. It means they read the code, and they make the bonds between the amino acids the same way, or almost the same way in all cells. They developed with evolution. They are much more complicated in human than in bacteria, but the main tasks in all cells. They read the code and they make the proteins in all cells. There is a huge number of ribosomes in each living cell. In mammalian, in the liver, sometimes in some liver, In some liver cells, there are 5 to 6 million ribosomes, all the time working. In bacteria, even in bacteria, that is an organism with a single cell, one cell, there are 80 to 100,000 ribosomes. Huge amount working all the time. They work fantastically. In vivo, it means inside the cells, they can produce 40 bonds. The bonds between the amino acids are called peptide bonds, 40 peptide bonds in 1 second. And they hardly make mistake, about a mistake to a million. Now about myself, I was a student, a chemical student. I had to make 1 bond. I was the fastest in the class because I was poor and I had to work in many jobs, so I had to finish my lab assignments the first or very fast. So I was the first and I finished 1 bond, which I had to make. They make 40 in 1 second. You know how long it took me to make 1 bond? 6 hours. And now chemistry developed, and it needs only 2 hours for 1 bond in the lab, but not 40 in the cell like the ribosomes make. So you understand why I was so interested to know how they do it. It's a cellular factory. So here I can show them as a factory, now that we know how they work. They have 2 floors. The top floor is for reading the code, and the lower floor is for making the bond. So what happens? The code comes in and is being read here. And when a triplet comes here, it looks for the amino acid that is coded by this, and this amino acid will then come inside the lower floor and connected to the growing protein that comes out from the lower floor. Then this truck that brings the amino acid, which is also a molecule, an RNA molecule called tRNA, will become empty and will go out with the empty trucks. The code will progress in 1 codon, and everything needs really very little energy. So what actually we see is the ribosome is made of 2 subunits, the top floor and the lower floor. The top floor is small subunit. The lower floor is the large subunit. They are independent, they live alone in the cell, and they come together to make a functional ribosome only when the cell wants. You will see it in a movie in a minute. But when they come together, they associate very accurately. The lower has the position where the decoding happens, the decoding center, the upper, and the lower has the position where the bonds are being made. And what connects them are these tracks, the tRNA molecules. So when we got the structure, we wanted to show it in a movie. I hope it works. Yesterday it worked. And we made a movie together with 2 students of the Art Academy in Jerusalem, which shows how the bacterial ribosome works. Yeah, it comes. It comes. So what we see here is the small subunit and the code. The code is reaching it, and it will take it. And to the small subunit, there is here a factor, a non-ribosomal factor, cellular factor called initiation factor. In bacteria, there are 3 of them. Number 3 is sitting here, the blue thing. Look how it monitors the entrance of the code into the path in the small subunit. So this is the factor. Now, when it is happy, the first track can come in, the first tRNA, to its position, and it's also brought by initiation factor. It's number 2. And they are here now, and when they are happy, they go. When everything is fine, they go away. And the large subunit can reach, associate, and make chemical bridges, and now the active ribosome was made. And we look at it from the solvent side, the large subunit on top, the small subunit below. More and more trucks are bringing amino acids with elongation factors. They go in, they are being read here, decoded here, and they make the protein here, and the ribosome helps in and out, 40 times a second. So now we took away the large subunit so that you can see the decoding happening on the small one, and the bond is made here, and the newly born protein goes into a tunnel, like a pipe that is sitting in the large subunit, spans it, and finishes at the end of the large subunit. And the process continues as long as there is a code, more and more amino acid come in, and the protein grows and starts folding. And when there is a codon says stop, the thing is finished. Instead of elongation factors and new amino acid, there is another factor, recycling factor or release factor. It goes in, this yellow thing. It lets the protein come out. All the amino acids, all the tRNAs can go and look for a new job. Also the ribosome, it released and it dissociated the two subunits, and everything else is now looking for a new job, and the newborn protein can do what it needs. That's all. This is the whole story, ladies and gentlemen. So, just to show you that we know the position of every component of the ribosome, there are in the ribosome 2.5 million atoms. They are arranged in two types of compounds. Also RNA, ribosomal RNA, which is shown here in gray, and many ribosomal proteins, 54 in bacteria, that are shown in all types of colors. What you see here rotating is the small subunit and the large subunit and their composition. And what I show now is what they do. The small subunit does the decoding, and the large subunit, the bonding. This is the ribosome, the bacterial ribosomes. Now many more ribosomes are known, but this is a good representation of the bacterial ribosomes. All the functions of the ribosomes are done by RNA. This is the main component here. You can see 2/3 of the ribosome. The ribosomes are not normal protein enzymes, they are RNA enzymes called ribosomes. Both activities are made by the RNA, the decoding and peptide bond formation. Now let's talk about something related, and these are these people. You see they are all either composers or philosophers or writers. They are giants. They are fantastic people. They are above and beyond everything. They are intellectual giants. What is common to all of them? They all died young, very young. All died before or at the middle of last century. The last one is 1950, Orwell. Others in the 17th, 18th century. What happened in the middle of last century? Why did they die? They all died before then from infectious diseases. In the middle of last century, there was the finding of compounds that can take care of infectious diseases called antibiotics. They came into use in the middle of last century. The first one that was discovered and used is called penicillin, and what it does, it chops the walls of cells of bad bacteria. But actually, most of the antibiotics are stopping protein biosynthesis because it's such an important process. And most of them are stopping or paralyzing the ribosome. So because of the fundamental role of it, almost half, over 40% of the antibiotics are paralyzing it. How do this very little antibiotics work. First of all, what are the natural antibiotics? These were the first to come into use. They are the weapons that bacteria from one type is using to interfere with the life of the other types, other species. How do they work? They are very small, and they have to paralyze the huge ribosomes. It's 2.5 million instead of, 500 against 2.5 millions. They do it by binding to the functional sites. So what you see here is the skeleton of the large and of the small subunit, and the circles show where they come in, where the antibiotics bind, and you can see that all of them bind to functional sites, either the decoding here, or bonding, or in the tunnel, or hinging these motions. And instead of talking, I will show you again a little video that explains it, that shows some examples. Good. So here it is. This is a very small one called adeine, and it binds to the small subunit and doesn't let the code progress, the code bind into its path. You see, that's all. It's a barrier. You just have to know where to be, but otherwise, this small one can stop protein biosynthesis. Second one is tetracycline, which occupies the position of the second tRNA. In order to make a bond, you need 2 of them. So here it is brought, but it cannot bind because its position is taken. That's all, tetracycline. Now I will show you 2 that act on the large subunit. First that I show is erythromycin. Erythromycin, it's the first one that worked on ribosomes. It sits in the tunnel for where the proteins come out. So only a very short protein can be made and then blocked. And the last one that I will show is the cleverest, is one that understands the chemistry. From chemical point is the cleverest. It goes into the bond. Look, look, look. Look, into the bond. That's it, no bond, no protein. Isn't it amazing? Bacteria against bacteria. Not big companies, not human. Bacteria against bacteria. Okay, bacteria have weapons, one against the other, and we are using the weapons. And the bacteria that are hit by the weapons try to resist it. So actually resistance to antibiotics is one of the most severe problems in modern medicine, and I'm sure you know it. Resistance grows because of more and more use, but in fact, resistance is a natural process of bacteria that being hit by other bacteria and they resist it. So I won't go to read everything, but I'm telling you. The increasing appearance of multi-drug resistance, even some bacteria can resist several types of drugs, together with the hardly any number of new antibiotics by the big companies, is becoming a real big overall over the world problem. And even the World Health Organization said that we are reaching post-antibiotic era. And the World Bank, estimated that up to almost 4% of the global economy will be lost by 2015 because of resistance, if nothing else is done. Yet, very few new antibiotics are made by the companies, and this is because there is a big distance between the human need to recover and the profit, which from antibiotics does not, for the companies, is not sufficient. So we have to do something. But first of all, we have to think, can we fully combat resistance, fully eliminate it? In my opinion, it's unlikely, because bacteria want to live, they will find ways to live, and because they are cleverer than us in terms of survival. So we have to find ways that their cleverness will not be so pronounced. Yet, most pharma companies don't make them, as I said earlier. I just want to show it in figures. So this is the way the resistance grew in pathogens of 3 different bacteria. You see how huge slope up. So in 2002, they really reached huge numbers. But the companies, they go down. And in the whole period between 2003 and 2007, there were only 2.5 new compounds. That's not the way that we will fight. So we decided to do some research in my own laboratory, very modest, a small laboratory, but we got really nice results. So until now, all what we knew about antibiotics binding to ribosomes is using ribosomes that are not pathogens, that are not disease-making. And from this, we learned where the antibiotics bind, like I showed you in the little clip. By the way, the 2 clips are available in YouTube and available for a long time now. But this, understanding the common factors is very important, but it doesn't explain the specificity of species. Therefore, we focused on one of them. We grew crystals. I won't talk about it now. I'll just tell you what are the lessons. All bacterial ribosomes are similar. Doesn't matter if the ribosomes are from good bacteria or bad bacteria. This is general lesson, and I can show it. Here is a small subunit, the large subunit of the pathogen, the one that makes diseases, and it's very similar. Even the active sites are very similar to what you showed earlier. So this was the general lesson. The first specific lesson is that it's still possible to make better antibiotics. We are collaborating with a little pharma factory in Vienna, and they could make, it's called Nabriva, and they could make 25-fold better antibiotics. I will not talk about lesson 2 and 3. I will focus on lesson 4 because this was beyond my expectations. It's the most exciting result that I didn't even expect that we found. So let's have here a look at one of the ribosomal proteins in many bacteria. And we aligned the sequence of the amino acids. And here you have a table that shows that blue is the most different and red is the most common. And look, most of them are common, or most common, or at least very common, except for this part, which is specific to this bacteria. And this bacteria is a pathogen, a bad bacteria. Only it has it. So this is so specific. Maybe we can target it. We can hit it. You can see another one, the same story. In blue here, cyan, we show the RNA, the ribosomal RNA, that is exactly the same in these 4 bacteria, 2 pathogenic and 2 not. They can also show one of the proteins. It's called L17. It's the same in all 4 bacteria except for here. And this is specific to the pathogen and can be a new potential target for antibiotics, and it will be specific. So some people, especially scientists, chemists, say that proteins are not such good targets because they can move and it's flexible. So we looked at the RNA. And what you see here rotating is the RNA of the large subunit, the normal one, not pathogenic, in gray, and these pieces are addition in the pathogenic. From the point of view of statistics, they were called standard deviations. The statistic people didn't really look at them. But when we focused on their structure, we saw something really interesting. So now let's focus on this part. And here it is. You see, until here, everything is the same, but this is addition. Here is the addition. Until here, everything the same, undistinguishable. But the addition, maybe statistically is not important, but structurally it's fantastic. It can be a new binding site, which is specific to this pathogen. We could see in other places too, and we could actually identify 25 new potential unique sites like this, and 16 of them could be used for blocking, for inhibiting protein biosynthesis. And these sites are not involved in the primary ribosomal activity, not in decoding, not in bonding. They are involved in other interactions that are needed for protein biosynthesis in the cell, in talking to cellular components. But the ribosomes, the bacteria didn't understand it yet. They are clever, but not so clever. No pathogen contains genes for this place, for modifying these places. So we can use them. And we can develop for them antibiotics that will be specific and degradable and environmental friendly. So here, I didn't talk yet on specific and also not on environmental friendly, and let me say a few words about it. Specificity first. Do you know that there is a body of bacteria called microbiome? This is inside us, inside every living organisms. These bacteria are residing in the flora of semi-exposed organs like the gut, the ears, the lungs, the skin, the nose. And they are harmless, and they are very, very important to our life and well-being life and our health. There are trillions of them in every living cell, and they have the structure of normal bacteria. So normal antibiotics can damage them, and sometimes they do if they can penetrate to these places. So this, we want to be specific and not to disturb these ones. Also, environmental aspect, environmental consideration. Most of the antibiotics that are used now have small pieces in them. You can see here some examples that are not digestible. So we get them out of us, and they are then not degradable. So then they can, when they are in the sewage, they are so small that the purification facilities not always catch them. Sometimes they find a way to sneak out and get into the water that is used for irrigation. And then we can get them back via the grass or the milk or the flowers or the trees or the fruit or the vegetables that grow on this water. And they come back to us and increase resistance. And actually we don't need them. So in our new ones, the ones that we make in positions similar to this, we can chemically design them so they are not so disturbing to the ecology. They are eco-friendly. They can be degradable. They can have chemical properties that can be optimized in terms of toxicity, or it means, minimized in terms of toxicity, optimized in their antibiotic action. And they can be specific because we target them only to bad bacteria, to pathogens. So this can be used for these things. And in summary, our insights told us about what happens in pathogens, for suggesting novel sites, I just showed it, for suggesting how to improve chemical properties of non-antibiotics, I didn't show, but I said, for better distinction, I hinted at that, and for making pathogen-specific antibiotics, like personal medicine, but in antibiotics. So this is our dream for the future, and it all started because I wanted to understand how the ribosome works. So for years, people say that I will not be successful, and they call me the dreamer or the fantasionaire, but the Weizmann Institute, especially its president, allowed me to be there. He was a biologist, but when he was replaced by a physicist, they still let me work on my dream, and they were encouraged by the Scientific Advisory Committee. The whole study, the whole work started with a very strong collaboration with Max Planck in Berlin, especially its director, Dr. Wittmann, who really wanted to know all about ribosomes and helped, was very important in setting up a specific research unit for us in the synchrotron, in the big facility in Hamburg, where this was the first synchrotron, this accelerator that was used for biological structure determination. He died 10 years before the structures came, and then was replaced by Franceschi and Fruzzini. So I had two groups, one working in Hamburg in Germany and one in Weizmann in Israel. And I want to thank the members of these groups for their devotion, determination in good times, but more in bad times. So this is the group in Hamburg that was terminated in 2004, and it had angels, the two technicians that said it's not possible to work on ribosomes unless you are an angel. And the group in Israel that is still functioning, you can see your members, some of them are already left for the last 3 years. I want to highlight an Indian postdoc, a fantastic postdoc called Tanaya Bose, who got her baby last week. And let's talk a little bit about this, a few minutes. And what I'm going to say now is about female, to the girls here, girls that want to go to science, and the society or the family say, no, it's not good for a girl to be a scientist. She cannot be then a good housewife. So I want to tell you that's not correct. Dr. Anat Bashan, who is the senior scientist of the group, she really runs the show, not me. I'm here only for decoration. She's doing the thing. She's a fantastic scientist, and she has 3 children, all fantastic, and a great husband and a great home. So it's possible to be a very good scientist and a very good mother. Ella is also fantastic, but she has only 2 children. Well, maybe that's enough. She's a great mother for her 2 children. Raz is one of the geniuses. He has 3 children, but he doesn't belong. He's a man, so let's go to the next one, Tamar. Tamar came to us 16 years ago as a very, very young student for 10 weeks. She's still around, 16 years. Now she has 3 children and everything. She's married, she has a home and so on. And at the day that I took the picture, she had her birthday, and she baked a cake. Can you see the cake? Maybe you don't see well enough. So here is her cake. And this shows that in my group, ribosomes are considered sweet. And I also want to thank my family. I cannot show my mother, my sister, and everybody. I just show my daughter, who is an MD, and her daughter, my granddaughter, who at that time was a school kid. And they supported me all the time, when I was there and when I wasn't there. They were always very, very friendly and supportive, and I thank them very much. And I just want to show that it's possible even to be a grandmother and scientist. Look what my grandchild said 10 years ago in Paris when I got a prize. She said, my name is Noah, Ada's granddaughter. She gave a speech. As you know, it means me, is a very busy scientist, but she always finds time for me. That's what she felt as a young 10 years old girl. And she gave me a prize. That's not her prize. This is the Nobel medal. It's very important. But her prize is at least as important for me as this one. And her prize is, the grandma of the year is E Yonath. And when I asked her which year, and said, which year? Every year. You have to reprove yourself every year. And if you fail, I take it off the wall. And I want to show you, ladies and gentlemen, it's still on the wall. So I'm still the Grandma of the Year. Ribosomes became really popular in Israel, in a party, in a carnival. He's me and his wife. Look at his wife, she's dancing here, and protein comes out of her. She's the ribosome. And this is what happened to me. An artist drew me as head full of ribosomes. If you look at it, small subunit, large subunit, small again, there is a symmetry here. Again large, again small, and he thinks that antibiotics could go here and here. Thank you.
How can the antibiotic differentiate between bacterial ribosome sites and human ribosome? How do they specifically attack pathogens in a human body?
So the distinction between bacterial ribosomes and human ribosomes is based on subtle differences, very, very small. I couldn't go here to explain it, not only because of the time limit, but also because of the knowledge that has to be known. But now that I really wanted just to understand the structure, I didn't even dream about understanding ribosomes. Now we understand all antibiotics. I didn't even dream about that. And we also found out how they distinguish very small molecular changes, very, very small, minute, sometimes only a few atoms. And it's all explained in Google.
Once again, thank you very much for your time, and big hand for